Welcome to Excellent Grades Academy. This is Dr. Byson EM. Today we are looking at histology, and under histology we are looking at the lymphatic system, which is histology lab practical number 12. You can register with us for all medical courses on +260 975 497790. Let's get into this lab. Okay, so what are our learning objectives for this lab? Number one, to identify the main lymphatic organs. You know that your lymphatic organs, lymphatic organs are classified as primary organs, which are the bone marrow, and the thymus. So the thymus is the lymphatic organ here, not really the bone marrow. Secondary organs are the lymph nodes, the spleen, the tonsils, and the lymphatic nodules. So in this lab, we're going to see the histological features of the lymphatic organs, which are the lymphatic nodules, the lymph nodes, the spleen, the thymus, and the tonsils. Number two, you give the detailed features of the lymphatic organs histologically. Number three, to discuss the location where they're found and specific functions of these organs. Number four, to discuss the development of the thymus, the spleen, the tonsils, and the relevant developmental abnormalities. Fifth, and not the least, name the common pathological conditions resulting from the malfunctioning of these organs. Okay, so we're going to look at the spleen, we're going to look at the tonsil, we're going to look at the thymus, the nodules, the lymphatic nodules, and the lymph nodes. When we look at all these, then our discussion for this histology lab would have been complete. Now let's look at the histological features of the spleen. So under the histological features of the spleen, we're going to focus on the lymphatic nodules. So label the lymphatic nodule, label the capsule, and also note the connective tissue trabecula. Also note the central artery that is in the lymphatic nodules. All right, so now at high power, you identify the scattered lymphocytes, the macrophages, the plasma cells, and the ordinary elements of the circulating blood. Also, the reticular framework of this tissue cannot be seen by this framework. Now, in the exercise, you're going to define or explain what is meant by the white pulp and the red pulp. And then note the differences in the organization of the spleen and the bone marrow, something that we're going to talk about. For the slide of the spleen, only draw it at high power. Okay, so this is a histological slide for the spleen. What you see, the outer part here, this is a capsule. So what is labeled here, this is what we call the capsule. Okay. Now, this capsule is going to give, so this is a capsule, is going to give layers of connective tissue that are going to go in the spleen. These are what we call trabeculae. So what you see here, this is trabecula. So trabecula. Okay, so that is a trabecula. This is the trabecula also as well here in longitudinal section, and this is also the trabecula in cross section. Okay, the red substance that you see here, this, everything that appears here, this is what we call the red pulp. So this is called the red pulp. The red pulp has got red blood cells. That's why it appears red. So it has red blood cells and cells that are called macrophages. So you know macrophages from your immunology, these are white blood cells. So the red pulp has got red blood cells and macrophages. What you see here, they appear like nodules. Okay, so these are nodules. These are what we call lymphatic nodules. These lymphatic nodules are found in the white pulp. So this is a lymphatic nodule. So lymphatic nodule. The lymphatic nodule, as you can see here, it appears purple, but on the center it's pale. So the center is what we call the germinal center. Okay. So the center is the germinal center. Okay. Now the question would be, why does the lymphatic nodule appear dark in the peripheral and white on the center? This is because on the peripheral part here, you have lymphocytes that are not actively dividing. And these lymphocytes that are not actively dividing have got a very large nucleus that can retain stains. Okay, so the lymphocytes on the peripheral side here are not actively staining, but the lymphocytes that are on the center here are actively dividing. They're actively dividing. So because they're actively dividing, their DNA in their nucleus is condensed, is in chromatin phase. So that's why they do not retain a lot of stains here. So this part has got dividing lymphocytes, and the peripheral part has got non-dividing lymphocytes. This is what we call the white pulp. Now, on the white pulp, on the nodule, lymphatic nodules, you find what we call the central artery. Okay, this central artery, you find it in lymphatic nodules in the spleen, but you do not find this central artery in the nodules that you find in the lymph node. That's one key difference. The other difference between the spleen and the lymph node is that the spleen is not divided into the cortex or the medulla. But as you are going to see, the lymph node is divided into the cortex and the medulla. So these are the parts of the lymph node, the spleen rather, that we want you to label. Want you to label these parts. Okay, let me show you another histological slide for the spleen. So this is the spleen at low power. So at low power here, you just observe. So just observe. Don't draw. Don't draw. So as you can see here, this is the capsule that is surrounding the spleen all over. And then this capsule is giving off this, because this is the capsule here. The capsule is drawn like that. This capsule is giving off connective tissue inside of the spleen that is called trabecular. And what you can see here is that you have nodules, the red pulp rather. So this is the red pulp, which contains red blood cells and macrophages. And then you can see nodules here. So these nodules are aggregates of lymphocytes. Okay, aggregates of lymphocytes, and they make up the white pulp. They make up the white pulp. What you can see inside here are just your arterioles. These are just your arterioles. All right, so this is at low power. You're not drawing anything at low power. This is at high power, showing us what we've already talked about. So this is a capsule here. The capsule gives off the trabeculae, connective tissue. So this is the trabeculae. And this is the white pulp, because it has the lymphatic nodules. What is surrounding the white pulp here, this is the red pulp. So we used another stain here. This is the red pulp. We already talked about the red pulp. We said the red pulp is made up of red blood cells, and macrophages. Okay, and macrophages. The white pulp is made up of T lymphocytes. It's made up of T lymphocytes. Okay. And in the white pulp, we've got a central arteriole here. We said the central arteriole will differentiate the lymphatic nodules in the spleen from those that are found in the lymph node. Okay. Now let's move on and look at the lymph node. This is the spleen still. Okay. So I put several diagrams here so that you can see where the red pulp is, where the white pulp is, the different pulp, the parts of the white pulp, and the arterial that is found near the white pulp. Okay, so you can use any of these diagrams to draw this at high power. Remember, you're only drawing the spleen at high power. Okay, now let's look at the histological features of the lymph node. So the characteristics that you should look for the lymph node is that you should identify the cortex that has got numerous lymphatic nodules and the central medulla with the irregular medullary cords and sinusoids. Okay, so also note the connective tissue that blend in with the surrounding tissue and sending trabecular or septa into the parenchyma. And then at high power, identify the subscapular or marginal sinus. Subscapular or marginal sinus. Now let's look at the lymph node. So this I put here, this is just a diagram, a schematic diagram that is showing your lymph node. Okay, so this, the green part here, this is the capsule. Okay. So now just below the capsule, you've got this space here, this white space here. This white space here is what is called the subscapular sinus. Subscapular sinus. Now, lymphatic drainage, these vessels are afferent lymphatic vessels, meaning that they are bringing lymph from the interstitial spaces into the lymph node. When the lymph comes here to enter the lymph node using these vessels, which are lymphatic vessels. The lymphatic vessels, the lymph will now pass through the subscapular spaces and it will enter the trabecular sinus, which is this sinus here. From the trabecular sinus, it will go into the medullary sinus. From the medullary sinus, it will now exit the lymph node through the efferent lymphatic vessels. Okay. Now the parts of the lymph node that you're supposed to know, the capsule is the green part, the outermost part. And then underneath the capsule, you have the subscapular space. And then from the subscapular space, you've got the cortex. So this is the cortex. Okay. The cortex is divided into the inner cortex and the outer cortex. So this is the outer cortex, just immediately after the trabecular sinus, the subscapular sinus rather. You have the outer cortex, the inner cortex, and then you've got the medulla, where you find the medullary sinus. Okay, so this is the orientation of the spleen. Let's look at it on a histological slide. Okay, so now this at low power is a spleen. What you see here, this is the capsule. As you can see, this is a capsule. Then the capsule will send in trabecular. So this is trabecular. The trabecular is making these spaces, as you can see. These spaces are trabecular sinus. So here, this is the capsule. This is the trabecular. Trabecular. This is the outer cortex. Cortex. And then this is the inner cortex. Cortex. And where you find the light space here, this is the medulla. This is the medulla. Now, as you can see, the outer cortex, let me get it from here. The outer cortex has got lymphatic nodules. Okay. It has lymphatic nodules. So they're not very prominent here, but they're there. So the lymphatic nodules are these nodules here. So the lymphatic nodules are simply aggregates. They're simply aggregates of lymphocytes. Okay, lymphocytes. Now, one thing of note is that the lymphatic nodules in the lymph node do not have the central artery. That's the differentiating factor between the lymphatic nodules in the spleen and the lymphatic nodules in the lymph node. And as you can see, the lymph node is separated into the cortex and the medulla, while in the spleen there was no division like that. So that's one other difference. So at low power, this is what we can see. Okay, that is what we can see at low power as well. So this is a capsule here. This is the inner capsule that is sending trabecular, capsule, trabecular, capsule, and then you've got these nodules. You've got a nodule here, 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 you've got a nodule here. These lines, these are trabecular sinus. Okay, trabecular sinus, the ones we saw there. And then this trabecular sinus, you see there are a lot. They're going towards the medulla. So this part here, which is light, this is the medulla. The medulla. And the spaces that you can see in the medulla here, these are medullary sinuses. So medullary sinuses. So this is what you are supposed to label when you draw it at low power. So this is the low power. All right. So this is high power. It's supposed to show the subscapular marginal sinus, but this diagram is not very clear. Okay, it's not very clear. But if this is the capsule here, okay, this is the capsule, this is the capsule. Okay, so this is the capsule here. This space that you see here, that I've shaded here, this space is the subscapular margin or subscapular space, subscapular sinus. It is just it's just between the capsule and the cortex. So the space between the cortex and the capsule, that is what we call the subscapular sinus. So this is the subscapular sinus. From the name itself, you can tell sub means under, capsular, so it's subcapsular. Sub means under, capsular is from capsule. So under the capsule there's a sinus, which is a space. So draw this and show the subscapular sinus there. So this is what you are going to see in the lymph node. Okay, that is what you're going to see in the lymph node. Okay, all right. Let's move on to the histological features of the tonsils. Okay, so under the tonsils, on the free surface is a familiar epithelia. What type of epithelia is there? Which is invaded by the lymphocytes. Okay, so from the capsule, you have extended tonsillar crypts containing some mucous material. Note the numerous lymphatic nodules, which have a well-defined relative germinal center, and some connective tissue trabecular with blood vessels may be seen. So now let's look at the tonsil. So this is a tonsil. So the tonsil is here. The tonsil is lined by this capsule here. Okay, so this capsule is lined by stratified squamous non-keratinized epithelia, and it will form a crypt. Okay, so this crypt here is dividing the tonsil into lobules. This crypt is dividing the tonsil into lobules, and then these lobules here, they are lymphatic nodules here. So this is a lymphatic nodule here. This lymphatic nodule has got a lot of cells here, which are just, so this is a lymphatic nodule, which contains aggregates of lymphocytes. So it has aggregates of lymphocytes. So make sure you draw and show the crypt, and then make sure you draw and show the lobules. So these are the lobules here. You show the lobules, you label the lobules, and say the lobules have got aggregates of lymphocytes. And some lobules have got a germinal center. So if this is a lobule, you can see here that on the peripheral it is dark. Okay, so in this peripheral it is dark like this. This dark periphery has got non-dividing lymphocytes, but at the center here it is light. This center is called the germinal center. The reason why the germinal center is light is because it has dividing lymphocytes. That's why in a lymphatic nodule you may find some lymphatic nodules have got a dark peripheral center and a central zone that is light, which is called the germinal center, meaning that the peripheral zone has got non-dividing lymphocytes, and then the germinal center has got dividing lymphocytes. Okay, now if you look at this, this is simply a blood vessel. So this is a blood vessel here, and this is just connective tissue. Okay, so this is connective tissue that is separating the tonsil into lobules. All right, so this is a lingual tonsil. Let's look at another histological diagram. This is it. Okay, so you have a capsule here. Okay, this capsule forms a crypt here. This crypt is dividing the tonsil into lobules. These lobules have got these lymphatic nodules. You've seen this lymphatic nodule has got a germinal center here. This lymphatic nodule again has got a germinal center here. This lymphatic nodule has got a germinal center. This lymphatic nodule here, germinal center. So draw them like that. Lymphatic nodule has got a germinal center. Lymphatic nodule has got a germinal center. So as you can see, the tonsil has got a lot of lymphocyte aggregates that are termed as lymphatic nodules, which are secondary lymphatic nodules because they have germinal centers. Germinal centers have got lymphocytes that are undergoing division. Okay, now let's look at the histological features of the thymus. The thymus is a primary lymphatic organ. So it should note the connective tissue septa carrying the blood vessels and separating the organ into lobules. And there are no lymphatic nodules here, each with a darkly staining cortex and a lighter medulla, in which Hazo's corpuscles are seen. And at high power, note that the densely packed lymphocytes, which are called thermocytes, in the cortex, and the large epithelial reticular cells in the medulla. Examine the Hauser's corpuscles at high power and note their structure. Okay, so the thymus, we're going to look at the thymus in children and the thymus in adults. You know that the thymus in adults involutes, or it dies off. So what you're going to see is that the thymus in children is going to look very complete and healthy, and the thymus in the adult is going to be involuting. So this is the thymus in children. Okay, so here, this is a capsule. So this is where the capsule is. So this is a capsule here, and then the capsule is giving off these are called septa. So these septa are dividing the thymus into lobules. So there's a lobule here, there's a lobule here, there's a lobule here, as you can see, even here, lobule. The septa is dividing it into lobules. Now, as you can see, this lobule has got this part here that is dark. This is called the cortex, and it has this part here that is light. This is the medulla. So this is the cortex of the thymus. This is the medulla, the middle region. Now the medulla has got these corpuscles, which are called Hazo's corpuscles. Okay, Hazo's corpuscles. Hazo's corpuscles. Okay, so it's very important for you to know those corpuscles. Okay, so now in the cortex we have lymphocytes that are called thymocytes. Okay, so this is at low power. Draw at low power. At high power, it's going to look like this. So this is high power. The high power, the capsule is this here. So this is the capsule here. Label it like this, capsule. And then this is the cortex. So this is the cortex. The cortex, we said, has got lymphocytes. These lymphocytes are called thymocytes. Thymocytes. Now, this light part here is the medulla. As you can see, the medulla has got the Hazo's corpuscles. Hazo's corpuscles. So use this diagram and draw it at high power. Label the cortex, the capsule, the medulla, and show the Hazo's corpuscles. All right. So that is just the thymus at medium power. You're just observing here. So here, just observe. Just observe. You've seen these are the Hazo's corpuscles there. The Hazo's corpuscles. Okay. Meaning that this is the medulla here. This is the medulla. And then this is the cortex. There. So this one you're just observing. Okay. This is also the Hazo's corpuscles. It has been magnified at high power. Okay. This is high power Hazo's corpuscles. Just observe here as well. This is the Hazo's corpuscles as well. Okay. All right. Now this is so the thymus that I was showing you here. This is the thymus for the children, the child thymus. The thymus that I'm about to show you here is for the adult thymus. So for the adult thymus, what you're going to see is that this is the cortex. And then this is the medulla. You've seen that it's involuting. Why are we saying it's involuting? Because the cortex is becoming adipose tissue and it is compressing the medulla. So the thymus is dying off. So draw this at high power. Okay. So this one here is the Hazo's corpuscles. Hazo's corpuscle. Look at how small the medulla has become. The medulla is being compressed. Okay, it's being compressed. It's very small. Why? Because the thymus is dying off. So draw this, label it adult thymus. There is no differentiation between the cortex and the medulla. So let's zoom in. You've seen. So the medulla is dying off. Okay. So this is the cortex. This is the medulla. By a certain age, the thymus will totally involute. This is showing the same thing. This is the only functional part. The entire of it has been involuted or is dying off. Okay. So now, that was the last histological diagram that we were doing. You can register for all the courses on +2609754977790. I hope you learned something in this histological presentation, and I'll see you next when we'll be doing cardiovascular system and the integumentary system.